Hey guys, so I just want to make a quick video reviewing this FRQ. So this FRQ is Unit 5, so it's meiosis and genetics. So if we look at it, we can read the prompt real quick. In a certain species of plant, the diploid number of chromosomes is 4. 2n equals 4. Remember, diploid means that an organism has two sets of chromosomes, one set from mom and one set from dad. So this 2 represents how many sets of chromosome they're receiving. N is the haploid number, and 4 would be this organism's diploid. We would be 2n equals 46. Okay. Flower color is controlled by a single gene in which the green allele, capital G, is dominant to the purple allele, little g. Plant height is controlled by a different gene in which the dwarf allele, capital D, is dominant to the tall allele, lowercase d. Individuals of the parental generation with the genotypes homozygous dominant and homozygous recessive were crossed to produce the F1 progeny. So we're assuming that this cell is representing the F1 progeny, and that would be the offspring of this homozygous dominant individual for both traits with an individual that is homozygous recessive for both traits. The first question is going to ask us to depict the 4 possible products of meiosis that would be produced by the F1 progeny. So we need to first remember what is meiosis and what's happening there. So remember, meiosis is the process. Okay, so remember, meiosis is the process of making gametes. And our gametes need to have half as much genetic material as our body cells so that when they unite with another gamete, then we're restored to our normal diploid number. So in humans, each of our gametes carries 23 chromosomes. And then it unites with another gamete that contains 23 chromosomes. And then we have a zygote that is going to have 46 chromosomes. So basically, it's a process of making 4 haploid cells from a diploid cell. So you're going to take a normal body cell that is going to divide twice to create 2 or 4 haploid gametes. So let's look at this. All right, so let's just look at this overview of meiosis. So remember, the first thing that happens in meiosis is our homologous chromosomes pair up. That means that the chromosome you get from one parent finds the same chromosome that you get from another parent. So we have 23 pairs, and so they find the ones that are corresponding to each other. So like, let's say right here corresponds for eye color in dad. Right here would correspond for eye color in mom as well. So they match up by size. And so we form those little tetrads. So first thing we're going to do is these 2 will match up, these 2 will match up, and these 2 will match up. And those are called the homologous chromosomes. After they find their buddy, they're going to all line up 2 by 2 down the middle. And we're going to separate that in half. So then we're going to have haploid cells that don't have 1, 2, 3, 4, 5, 6 chromosomes anymore. We only have half as much genetic information. 1, 2, 3. 1, 2, 3. Now, since these chromosomes kind of look like an X, they are in the replicated chromosome form. And we don't need replicated chromosomes. We only need one single copy of the information. Remember, replication happens during the S phase of interphase, and interphase happens before meiosis can occur as well. So we have replicated our chromosomes. So up here we have 6 replicated chromosomes. We've cut that information into half with 3 replicated chromosomes in each cell. And now we just need to separate the replicated chromosomes. So the next phase of meiosis, meiosis 2, looks a lot like mitosis, where all the X's line up down the middle, and then we just pull apart the sister chromatids at the centromere. And then we have individual unreplicated chromosomes in each of the cells. So we started out with 6, and we're going to end with 3. So that's why we know they're haploid. And instead of making just 2 like we do in mitosis, we make 1, 2, 3, 4. These are the gametes that can fuse together with another set of gametes to create a cell that would have 6 chromosomes again. So that's kind of what's happening in meiosis. And so again, we can look at this. Remember, during prophase 1, as the tetrads form, they can have crossing over occur. So this is what we did in class with the highlighters, where you had to highlight the crossing over, and it was really annoying, but that's how we get new genetic diversity. So it's really important to remember that crossing over is going to create new genetic diversity. And that's going to give us new combinations that weren't found in the parental chromosomes. So you might have a combination of like if this represents dad's eye color and dad's hair color or whatever, you could, and if this crosses over, then you could inherit mom's eye color, but still dad's hair color. So you can have new combinations of traits. It's not necessarily, or you can pass on new combinations of traits that were not in the parental chromosomes because they're shuffling the genes. So it's really important to increase diversity. And if we've gotten into evolution, we talk about how it's really important to have new, just different genes and different alleles and stuff, so that if there's diversity arises, things will be less impactful to a population if they're not all genetically the same. So if you're genetically identical to everybody in a population and something comes, it could wipe everybody out. But if people have special adaptations because of genetic diversity that allow them to survive, then they're going to be more resistant to anything that would come and harm the population. Okay, so then they line up 2 by 2, and then we're going to split that cell in half. And so this is that point where we're going to have 2 haploid cells, cytokinesis would occur. Then we have 2 haploid cells, and now they're both going to go through a process that looks very similar to mitosis, where they line up down the middle, and then they're separated during anaphase 2, and then they are completely split into 4 new cells by the end of cytokinesis. And again, these are all your 4 haploid cells. So that's just kind of a reminder of what is going on in meiosis. And so this question's asking you to depict the 4 possible normal products of meiosis that would be produced by the F1 progeny. Again, this is our F1 progeny. Show the chromosomes and the alleles they carry. Assume the genes are located on different chromosomes, and the gene for flower color is on chromosome 1. So the gene for flower color is on chromosome 1, and flower color was the G's. So let's see, what would the genotype be of this individual? Well, they tell us that they crossed a parent that is homozygous dominant with another parent that is homozygous recessive. So the F1 progeny, we can assume, is going to be heterozygous for both traits. And you guys could do the cross, but I think you can probably see that if you have one that's homozygous dominant crossed with one that's homozygous recessive, they're going to get one capital G from one parent and a lowercase g from another parent. And again, this one, the long one, is chromosome 1, so I'm putting the allele for flower color on chromosome 1. Now, they're also going to be heterozygous for tallness, and they're going to get one tall gene, or dwarf gene actually, and then they're going to get one tall gene. So the genotype of the F1 progeny, if we were to put it into letters, would be, hold on, I'm just gonna G, G, D, D. Okay, so that is the actual genotype of this individual. So what gametes can they produce? Well, they tell us that they are located on different chromosomes, so these genes are not linked together. So we can create the gametes from this. So the gametes arise from going through this whole phase of meiosis, where we would split the cell in half, and then we would go through meiosis again to have unreplicated chromosomes at the end. But if you guys remember when we did the dihybrid crosses in class, I taught you about the FOIL method for determining gametes when you have dihybrids like this, or 2 traits that you're analyzing. So if you remember, you're going to do the first combination of capital G and capital D, and the second combination of capital G and lowercase d. Then we could have our next combinations would be little g and capital D, and then we would have little g and lowercase d. So this gives us all possible combinations of the flower color and height that could arise when these gametes sort independently during meiosis. And so again, instead of having to diagram it all out, showing you how it splits in meiosis 1 and then it splits in meiosis 2, you can remember, well, this is how you're able to determine gametes more simply by just kind of doing this FOIL method. So that being said, I know my first combination could be, let's see, my first combination could be a capital G and a capital D. That is this combination. The next one could be a capital G and a lowercase d. And then my next combination could be a lowercase g and a capital D. And then lastly, my combination could be lowercase g and a lowercase d. Now, that just gives us the actual allele letters, but it told us that it wanted us to also include, to draw the chromosomes that they are on. So the chromosome 1 is the larger chromosome, and the chromosome 2 is the smaller chromosome. And so when we do this, we're going to show the larger chromosome next to the G and the smaller chromosome next to the D. Now remember, these started off as replicated chromosomes in the X form. And if you remember up here, these all start off as replicated chromosomes to begin with. But what do we have at the end of meiosis? We don't have replicated chromosomes anymore. At the end of meiosis, we have unreplicated chromosomes. So these started out with 6 and ended with 3 unreplicated chromosomes. So we started with 6 replicated chromosomes and ended with 3 unreplicated, or 4 cells that had unreplicated chromosomes with 3 chromosomes in each. So if we look at here, we start with 4 replicated chromosomes, and we're going to end with 2 unreplicated chromosomes in each. So I'm going to draw a large line, just 1, not 2. I don't want to form the X's because remember, these are unreplicated now. A large chromosome next to the G, that represents half of the X up there that would divide during meiosis 2. And then just the little short chromosome would be next to the allele for height. So that's what they want in those diagrams. They want to show all possible combinations of color and height, and they want to show the actual chromosome. And really they're checking, do you know that at the end of meiosis, they are unreplicated single chromosomes, and that you started with 4 in the original cell, that was the diploid cell, and you end with 2 in the haploid cell. So we have 2 chromosomes. That represents the haploid number. Because we started with 4, and now we have 2 chromosomes in each of the 4 cells. And then we've gone through all combinations that could happen between height and color. Okay, so hopefully that makes sense. That's what they're looking for there. And just to summarize that, you can look at your genotype, which would be heterozygous for both traits, do the FOIL method to determine all different combinations of G and D. Go ahead and put those different combinations in here. And then remember that at the end of meiosis, you would have an unreplicated chromosome for each trait. And so you just kind of draw the chromosome next to the allele so that you see what it would actually look like in the cell. Predict the possible phenotypes and their ratios in the offspring of a test cross between an F1 individual and one that is homozygous recessive. So the F1 individual, again, if you remember, is going to be the individual represented in the cell, which we said was heterozygous for both traits. And they want us to do a test cross. And a test cross is automatically going to cross with something that is homozygous recessive for both traits. So they told you the genotype of the test cross, which is generous of them because they could have just said a test cross. A test cross is so that you can figure out what the genotype is of someone that's expressing the dominant trait. So they use this a lot to determine if something's like purebred or not, because something can be expressing the dominant trait, but they could be heterozygous or a hybrid, not purebred. So if you cross it with someone that's homozygous recessive and all of the offspring show the dominant trait, then you know that the original parent that was expressing the dominant trait was homozygous dominant. But if you cross the one that's showing the dominant trait with someone that's homozygous recessive and you see a little bit of dominant trait and a little bit of the recessive trait, it means that the parent that was showing the dominant trait was actually carrying the recessive trait, and they're a hybrid and they're not purebred. So that's why we do a test cross. We really determine what is the genotype of the individual that's expressing the dominant trait. Are they heterozygous or are they homozygous? And so here we're doing a test cross between these two. We already know that the individual is heterozygous, so you can go ahead and add that genotype there. That was the same genotype that was represented by this offspring, and then they told you to cross it with that person. Okay, so remember, you could do the whole 16-box cross, but I recommend doing one cross for your color and then one cross for your height, and then we can determine kind of all of our different probabilities. Okay, so it says determine all possible phenotypes and their ratios of the offspring of a test cross between them. So what could happen from here if they're independently assorting, meaning that these are not linked together, and it's not like just because you're tall, you also have to be green. They say all combinations are possible. Well, we can have, what are our possibilities? We can have tall and, oh, not tall. Sorry. All right. First thing I like to do is write out both dominant traits. So our dominant trait was green and dwarf. So they could be green and dwarf. They could also be green and tall. They could also be purple and dwarf. And they can also be purple and tall. Okay, so we're going to figure out how many fall into each of these categories. So first thing I'm going to do is I'm going to cross my colors. So I picked the G's to cross because I don't want to do a giant cross. I'm just going to do each trait individually. And so I'm going to get this for my color. And then I'm also going to cross the height. Okay, so what percentage are going to be green and dwarf? So green is 1/2. The probability of them being dwarf is also 1/2. So our probability of them being green and dwarf is 1/4. All right, green and tall. Again, the probability of green was 1/2. Tall, the probability is over here, the 2 lowercase d's is also 1/2. So our probability of that being is also 1/4. Purple and dwarf is going to be 1/2 times 1/2. So we have 1/2 times 1/2. And that's going to be 1/4. And you can probably see that purple and tall is also going to end up being 1/2 times 1/2, which gives us 1/4. So all of them have the same probability. So our ending ratio would be a 1 to 1 to 1 to 1. And you would need to list all the phenotypes. So 1 green and dwarf to 1 green and tall to 1 purple and dwarf to 1 purple and tall. So that is your ratio. If the two, well, let me erase all this. Okay, so it says if the two genes were genetically linked, describe how the proportions of phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and the homozygous recessive. So basically they're saying this was your ratio that you got. How, if these genes were linked and they were not independently assorting, how would the proportions of the phenotypes of the resulting offspring be different? So how would your ratio be different if these genes were linked? So I'm going to pull up these sketch notes. Linked genes mean that these are genes that are very close to each other on a chromosome, and so they're likely to travel together. And so we have many more genes than we do chromosomes, so many genes that are located on the same chromosome. And when you're located on the same chromosome, if you inherit that chromosome from your parent, and those two genes are on the same one, then you're probably going to they're going to show up together. So because they're located on the same chromosome, they intend to be inherited together. So I think of this as like buy one, get one. So like if you have red hair, then you get freckles because they kind of travel in pairs. Now, most of our traits aren't linked. It's like just because I have brown eyes doesn't mean I also have type A blood. Like they're not linked together. That would be kind of weird if everybody who had brown eyes also had the same blood type. So it's not like that. But red hair and freckles just happen a lot because they're located very close to each other. Sex linked is a type of linked trait where we see things that travel together because they're located on the same chromosome. So things like boys have more color blindness is a linked trait. Some linked genes can be separated during crossing over, but genes that are closer together are less likely to be separated. So the reason we do see different things, someone can have red hair and no freckles, or they could have freckles and not red hair, or a lot of other linked traits could show you, is because crossing over can occur. It's just less likely to separate things that are really close together. So if they're like really close, it's hard for crossing over to separate them. So I show here that if this section crosses over with another section, then A and B are so close that they're probably going to get swapped out together. And so you can see A and B still travel together. So this one has 2 recessive alleles and 2 dominant alleles. And then when we swapped them, we still have 2 recessive alleles and 2 dominant alleles together. And that's just because they're close. It's hard to get separated during crossing over. Because A and B were close together, they were not separated during crossing over. And then these are the gametes that you could form. But these are still new combinations of traits between A, B, D, and E. And so we call these recombinant chromosomes, and these are parental chromosomes. Because linked genes do not assort independently, offspring tend to inherit more parental combinations than expected. So when we see genes that are linked, the offspring tend to look more like their parents than you would expect them to. And so we would expect in this situation, we expect to see a 1-to-1-to-1-to-1 ratio. Dwarf and green is the same amount as, so again, if this is green and dwarf, then we have the same amount of green and tall, and then we have the same amount of also, gosh, what color? Purple and dwarf, and then we also have the same number of purple and tall. So if these were linked, what could we expect to happen? Well, we would not see this ratio. We would not see a 1-to-1-to-1-to-1. We would see a different ratio than we would expect because the Punnett squares that we do use the assumption that we're independently assorting. So you could say that you would not see the 1-to-1-to-1 ratio. They would change, and I would say that they would probably look more like the parents. And so in the parents, we had one that was heterozygous for both traits and one that was homozygous recessive for both traits. So you're probably going to see more parental combinations than you do recombinant combinations. So you could say because if they were not assorting independently, or if they were linked, then they would have more parental trait combinations than expected. That would be a good answer there, is that they would have more parental trait combinations than expected, and they would not show the 1-to-1-to-1-to-1 ratio. There would be different combinations going on there. There'd be a different ratio. And then it says, and describe how the proportions of phenotypes of the resulting offspring would most likely differ from those of the test crop. Yeah, so I would say they show more of the parental combinations, and the recombinant combinations would come from crossing over. And I would just throw that in there because they like to hear that. So that is the answer to this. Again, it ties in meiosis and genetics pretty well together. Let me know if you guys have any questions. This is a short FRQ, so it's going to be worth less. It seems like it took a lot of work for me to get through it for it to be a short FRQ, but I wanted to make sure you guys understood the basis behind it. So anyway, hope you guys are doing well, and I hope this was helpful, and I will talk to you later. Bye.